Thank you and good evening, everyone. Within the next few minutes, I will present the results from our recent study, which was aimed at investigating the impact of dopamine on the brain signal and network dynamics during cognitive performance. Dopamine is known to underlie important aspects of cognition. Through multiple striatal cortical loops, dopamine innervates distributed cortical areas. One common finding is that individuals differ in the degree to which their behavior benefits from dopamine. Knowing that distributed cortical areas show coordinated dynamics which shape the functional connectome within the human brain, a question is whether and how dopamine modulates the functional connectome during cognitive performance, and whether this modulation can explain inter-individual variability in behavioral benefit from dopamine. Also, optimal cognitive performance is increasingly recognized to depend on the brain signal variability over time. And this has been shown that this variability is being modulated by dopamine availability in the brain. Therefore, another question is whether dopaminergic modulation of the brain signal variability is related to the dopaminergic modulation of the functional connectome. Thus, in our study, we asked whether and how dopamine modulates large-scale brain signal and network dynamics during cognitive performance. And in addition, we asked whether dopaminergic modulation of the brain dynamics can predict inter-individual variability in behavioral benefits from dopamine. And lastly, we investigated the direct relationship between dopaminergic modulation of the brain signal variability and dopaminergic modulation of the functional connectome during cognitive performance. To answer our questions, we conducted an fMRI experiment where healthy young adults perform a listening task once after receiving placebo and once after L-dopa. For the listening task, we use an auditory working memory task. On a given trial, first, the participant heard two consonant vowels, namely da and ge, and they kept them in memory. After a delay period, a cue was presented to the participant asking to allocate attention either equally to both consonant vowels or to selectively one of them only. And after a retention period and at the end of the trial, one of the consonant vowels were presented again, and the subject had the task to judge about the pitch of the consonant vowel retrospectively by responding as high or low. In order to investigate the brain signal dynamics and networks, we use a cortical parcellation scheme based on which we extracted local regional bold signals from the functional data. And then we simply use the variance of these signals as a proxy of information content. And then we continued by looking at the functional connectivity pattern between these signals using graph theoretical network modeling. To this aim, we use network efficiency, which is a network metric inversely related to the path length between brain nodes, and is considered as a proxy for parallel processing or functional integration within the brain. Now I would like to move on to the results. On the behavioral level and in line with the previous studies, we did not find a significant effect of dopamine on the behavioral performance. However, we observed a sizable inter-individual variability in behavioral performance under L-dopa in contrast to placebo. And this was the case for both response speed and perceptual sensitivity, or D prime. On the brain level, we started by looking at the brain signal variability. We found that under L-dopa, in contrast to placebo, there was a significantly higher signal variability in brain areas overlapping with temporal motor temporal and motor cortical areas. More importantly, we found for certain cortical regions, we found positive correlations between the modulation in brain signal variability on the one hand and behavioral modulations on the other hand. These regions overlap with supplemental motor area, inferior temporal cortex, and insula. On the functional connectome level, we found that under L-dopa and in contrast to placebo, functional connectivity was significantly higher, a relative hyperconnectivity. However, global network efficiency under L-dopa was significantly lower in contrast to placebo. That means a relative hypointegration. These effects were found within a wide range of network densities and were further supported by correlations with modulations in behavior. More specifically, those participants who showed higher increase in functional connectivity under L-dopa in contrast to placebo, they were faster. And those who showed stronger hypointegration or lower degree of global network efficiency under L-dopa in contrast to placebo, they showed higher perceptual sensitivity. And then we continued by looking at the cortical origin of the hyperconnectivity and hypointegration. We found that within cortical areas overlapping with a temporal single opercular network, nodal connectivity and network efficiency was significantly lower under L-dopa in contrast to placebo. However, in motor areas and visual cortex, functional connectivity was significantly higher. Lastly, we investigated the direct relationship between dopaminergic modulation of the brain signal variability and functional connectivity on one side and also nodal efficiency of the brain networks. And in this case, we found distributed cortical areas where modulation in signal variability under L-dopa was predominantly positively correlated with modulation in nodal connectivity or nodal efficiency. In order to conclude, the results of our study suggest that L-dopa increases brain signal variability and induces hyperconnectivity and hypointegration in the functional connectome during cognitive performance. In addition, the degree of L-dopa-induced modulation of signal and network dynamics proved successful in predicting inter-individual differences in benefits from dopamine. We also provide first evidence for a direct relationship between dopaminergic modulation of signal variability and the functional connectome. Overall, our study suggests that L-dopa enhances information processing capacity in the human cortex. This brings me to the end of my talk. I would like to thank my colleagues in the Auditory Cognition Lab, University of Lübeck, and the contributors to this work, and the funding organizations. And of course, the volunteers who participated in this study. And thank you very much for your attention.
The floor is open for questions.
Hi, thank you. This is a very good talk. I have a question about your subject. So you only perform the experiments with normal control. They don't have any disease. What if you do the same experiment, but your subjects are patients with Parkinson's disease, which is known for dopamine depletion? What would you expect to see?
It's a difficult question, and so first of all, so this would be because for functional connectivity analysis, you need to really control there are. I can point you to some papers where the authors argue that when it comes to comparing functional connectivity between control subjects and patients, you need to be cautious because functional connectivity already differs between normal and patients. But in terms of the direction of the modulation, I would expect that the patients would show actually lower functional connectivity in contrast to healthy subjects. But that would be just my prediction.
Okay, thank you.
Yeah.
Are there additional questions? I have one question for you. So I was wondering about what is the time scale of the effectiveness of L-dopa. So if you take L-dopa, how long-lasting will these effects be on the functional connectivity in the brain?
Yes, so we try to actually control for this or to make it as consistent as possible across participants. And there was, on average, like 30 minutes time lag between the onset of medication and the onset of a scan. But we try to make it as consistent as possible, and it should be based on the previous studies. It should be enough to actually should be short enough so that you can still observe the effect, the modulatory effect of dopamine on the brain dynamics.
But so basically, but would the effect last like across days or hours, or you'd
for
have
hours,
to...
For hours.
For hours.
Yeah.
Thank you very much.